This is a production of Cornell University.
So thank you, Lori and the department for having me here. It's my first time over to Ithaca. I really enjoyed my drive over yesterday. As I was telling Lori, I don't get a lot of time alone. And so the 6-hour drive actually was fantastic. And it was a beautiful day. I couldn't believe how warm it was when I got here. So yeah, so we're going to talk about Aspergillus today. I know perhaps maybe an organism that you don't hear a lot about here at Cornell. But I was a card-carrying plant pathologist at one time, and so I had fun pulling this paper. It was my very first paper I published as a graduate student. I worked on this in the late 90s using a technique that the students probably don't even know what it is anymore, random amplified polymorphic DNA markers, and to look at the genetic diversity of Fusarium exisperm isolates isolated from common bean and sugar beets in the central high plains region, which is Colorado, Wyoming, and Nebraska. So that was a big part of my graduate school work. And then I moved on and we did some functional genomics analyses in this really interesting interaction between Alternary brassicilla and Arabidopsis. And so what I thought was really exciting about this work was the fact that Alternaria was an actual primary pathogen of Arabidopsis, unlike early days, some of the systems that were being worked on at the time. And so we thought that was a nice introduction into how these brassica causing, these diseases of brassica plants were being mediated by the Alternaria genus. And so again, grad students, we use things called express sequence tags back then, which you probably don't know what those are either anymore, thankfully, because you can do things like RNA-seq now. So why am I here and what do I do? And as far as the students and postdocs go, I'm happy to talk about how I ended up switching into human fungal pathogenesis during my postdoc at lunch if you want to hear that story. But really, we ask very similar questions to what you ask when you're trying to understand how fungi cause disease of plants. And part of that reason is because antifungal therapies are not effective in a significant proportion of patients that have these invasive fungal infections, just like fungal pathogens of plants cause significant crop losses throughout the world despite use of antifungals and other management practices. So these organisms remain a huge problem. So kind of a big thing for our field. I mean, most of you, when you woke up this morning, you probably weren't thinking about getting an infection by a fungus. And when I first was introduced to that fact that fungi might cause disease in humans as a graduate student, when I made the switch, I was like, what, really? Like, it's just not something, fortunately, most of us grow up worrying about. And so recently, the World Health Organization, though, has recognized now fungi as a critical group of pathogens to study. And so in 2022, the WHO came out with their critical priority group. And so the organism that we work on, we're going to talk about today, Aspergillus fumigatus, is the mold. But you see Candida albicans and Candida auris, these are yeasts. And Cryptococcus is a fascinating fungus. It's a basidiomycete, but it also is a yeast. And so why did the WHO suddenly in 2022 become interested in these infections? And that's because medicine has changed and the world has changed dramatically with climate change, as well as advances in immunotherapy. And so many of the chronic diseases that we treat in the clinic now, the therapies we use to treat those diseases target the immune system. And so what we're seeing is an increase in infections caused by these fungi. But as plant pathologists, these fungi are everywhere. But yet we only, not only, but we see predominantly these 4 organisms showing up in these patients. So there's something unique about their biology that allows them to cause disease in a mammalian host. So just quickly, Aspergillus, it's like the screen's a little bit behind. There we go. A little bit of a delay. Aspergillus was discovered in 1729. I always like to talk about it's named after the aspergillum. If there's any Catholics in the audience, that's the holy water sprinkler. And so you can see the conidiophore, which is the asexual development structure of the organism, looks just like the aspergillum. He was a priest, so there you go, Aspergillus as the genus. And then fumigatus is Latin for smoke. And so this is a culture of Aspergillus fumigatus grown in the lab. And this green coloration here is the conidia. And it comes from a molecule called DHN melanin that actually happens to be a virulence factor for the organism. But importantly, those conidia are coated in hydrophobins. And those hydrophobins make the organism, the conidia, very hydrophobic, and so they disperse into the air. And so if you take a plate of Aspergillus and you just open the lid, you see smoke come off. And that's where it got its name, fumigatus, for smoke. It causes a pretty broad spectrum of disease. And so we're going to talk about invasive aspergillosis today, which is a systemic infection, but chronic aspergillosis is increasing in frequency now. And there's many manifestations of aspergillosis that relate to what's called a Th2 type response or allergy. So it's an organism that causes a very wide spectrum of human disease in a lot of different patient populations. So this is a CT scan from a patient at Memorial Sloan Kettering Hospital, not over in New York City. And it's from one of the patients of my collaborator, Dr. Tobias Hull. And what I'm trying to illustrate on this slide is that Aspergillus 1 is a pulmonary pathogen. And so in red, you can see one of these lesions forming in this patient's lung. And the crux of the matter is that our optimal antifungal therapies, which aren't that effective in vivo, have to target the organism in the right physiological context. And in our field, that's actually a significant challenge. And so how do you actually identify infection microenvironment relevant therapeutics? And a little bit of maybe a bit of a spoiler for later in the talk, but the way we've done this in our field for years, and as plant pathologists, you do this too when you go to inoculate a plant with an organism, is there's a big bottleneck here to get these morbidity and survival curves or plant death curves with our mutant fungus. This is the result you want to get, right? So you make a fungal mutant using fungal genetic approaches, and you hope that that mutant doesn't cause disease in your animal or your plant, and you get this beautiful survival curve, and you get papers and grants, and you've identified a drug target. But the problem with this is that often, what is often the case is to get a curve like this, you fail to initiate an infection. So these mutants tend to be very sick, and they are incapable of initiating that infection. And so in the agricultural setting, that could be very powerful, right? You could easily develop something you could spray on crops, for example, to prevent spore germination or prevent the development of an aprosoria that initiates the infection. But in a mammalian system, we can't predict who's going to get one of these infections. And so prophylactic therapies are a less viable alternative for developing new therapeutics for these diseases. So what happens is the individual gets an infection that's been established, and then we have to treat it. Okay? So we wrote a little piece on this, like just talking about the importance of these high impact virulence factors, which are important for initiation of an infection, but really unclear in almost every case because we don't really have the great tools to look at an established infection. We're not sure if they're disease progression factors because this is what you'd really want to target. When you get sick, you go to the doctor after you get sick. So we need to treat these infections in a different context than we often are studying them. So how we approach this in my lab, and so one of the ways is we've gone in to try and profile the infection microenvironment. This is an old experiment. It was one of the last ones I did as a postdoc with this idea in mind to try and understand what the physiology of the fungus was in an established infection. So a bit of a busy slide. It's a 1H NMR spectra. And so we have animals that were inoculated with just saline, and then animals where we give Aspergillus the canidia through the airway, right? So they just inhale the spores, they run around, they just inhale the spores. And then in the lung, what we did is you can actually get fluid out of the lung, called the BAL, bronchial alveolar lavage, right? And then you can take that fluid, and we did metabolomics on it. And we got a big surprise. And the surprise is these red arrows are metabolites associated with fermentation, particularly ethanol. So lactate, alanine, acetate, those are all metabolites that likely are coming from the host. So we can't rule out that the fungus is making those in vivo. But the ethanol we knew had to be coming from the fungus. And so this really was the piece of data that I started my lab on because the idea is that when do microbes ferment? Most microbes, okay? They ferment when oxygen becomes limited. And so you might be thinking, well, wait a minute, you're in the lung, you're talking about an obligate aerobic filamentous fungus. and you're telling me that it's fermenting and it grows in the absence of oxygen, or at least with limited oxygen environments. But I would argue that Aspergillus and a lot of our filamentous fungi that we study out in the soil, they're really good at recycling carbon and nitrogen. For most of them, that's their ecological role. And in compost piles, it's the #1 place where we could go out and get environmental isolates of Aspergillus fumigatus. When you turn over your compost pile and you see the smoke come out, the spores, a lot of those are fumigatus for a couple of reasons. One, it's thermotolerant. It's one of the few thermotolerant filamentous fungi. It can survive up to temperatures of 70 degrees Celsius. It can grow in the lab at temperatures at 55 degrees Celsius. Very rare for the molds. Secondly, if you go to your garden store, what do they try and sell you? They try and sell you devices to oxygenate your compost to speed up microbial metabolism to degrade the compost faster, which tells you that composts become oxygen depleted very rapidly due to microbial metabolism. So Aspergillus, it turns out, also grows even though it's an obligate aerobe, it will not grow anaerobically, but it can live anaerobically. It loves to grow in these low oxygen environments, okay? So what we ended up finding, at first no one believed us, but the ethanol data, we went and showed them. We used a tool from cancer. Okay, so in cancer, hypoxia or oxygen deficiency is a really important part of the tumor that confers drug resistance to a lot of cancer therapies. So as tumors develop, they become hypoxic in the middle. So there's tools to investigate that. So we showed this is a lung. Here's an airway. The Aspergillus came in through the airway. The green is Aspergillus growing out in the lung tissue. And the red now is showing you a region of the lung that's deficient in oxygen. Okay, so it's called pimidazole hydrochloride. It forms protein adducts on thiol residues only when oxygen levels drop below 1.5%. So at the site of infection, just like in a compost pile, the organism is having to deal with these low oxygen environments. Okay? So an important point here is that hypoxia was recently awarded the Nobel Prize for how mammals, another obligate aerobe, how they respond and sense oxygen levels through the HIF-1 alpha pathway. Really beautiful science. It's a really cool mechanism related to prolyl hydroxylase, which uses oxygen as a cofactor to hydroxylate HIF-1 when oxygen is abundant, and then when it drops, you don't get the protein hydroxylation, and the transcription factor can go activate the hypoxia response. But, and I think this is relevant for plant pathology, is oxygen demand, hypoxia occurs, the physiological response, when oxygen demand exceeds the supply. So that can occur at any oxygen level. And so when different fungi induce a hypoxia response really depends on their oxygen requirements and the oxygen levels in a given environment. Okay, so something to think about. And so we studied this for years, a lot of papers. We really got really lucky. We found this transcription factor called SRBA that, as you can see, when you make a null mutant, it cannot grow in hypoxia. It's the dream result. It's why I'm still here. It's why I ended up getting a job. This gene I love so much. We continue to study it in different ways. And so, it abolishes hypoxia fitness. It completely abolishes virulence of the organism. The nice thing here, am I showing you the data? Nope, not showing you the data, but this actually does initiate an infection, this mutant, but then once the hypoxia sets in, then the mutant stops growing completely and the animals are all able to survive. So we actually do have a project. It's kind of our latest paper looking at how to target this pathway therapeutically. It was a gift that kept on giving because the azoles are the main antifungal that we use in the clinic, just like in agriculture for antifungal activity. You can see that the Aspergillus wild type strain here is completely resistant to azoles, but this azole, voriconazole, it's susceptible to, so that is the primary therapy in the clinic if you get one of these infections. But our mutant was susceptible to both fluconazole, which is used to treat yeast infections, and then the azole. So virulence, drug susceptibility, it was the best gene we were lucky. Okay, and just to throw, again, that seems to happen in plant fungal pathogenesis as well. So this is the fermentation pathway in Fusarium that was shown by a group in Spain to be really important for causing Fusarium wilt, which is exciting, which suggests that, think about it. So are the roots full of oxygen? they're actually pretty oxygen deficient, especially when they get waterlogged. And so a lot of these root pathogens in particular must have to deal with low oxygen environments and able to thrive within a plant. And then in Magnaportha, Wong Hung Lee's group actually ended up studying SRBA in Magnaportha and show that Magnaportha, as it penetrates through the aprisorium, also experiences an oxygen deficiency. And it was kind of fun. I was at a GRC once, and Nick Talbot was giving one of his amazing talks, looking in the tissue, watching Magnaportha invade, and then he was losing GFP signal, and he couldn't figure it out. Like, and I went to him, I said, "You're losing oxygen as you get into the plant." And so anyway, really kind of cool. GFPs requires oxygen to sense. So that was all fine. I could talk to you all day about SRBA, but we wanted to find we had evidence to suggest that there were other regulatory pathways that were dealing with the response to oxygen in the fungus. And so, you know, I've talked to quite a few people this morning about RNA-seq, and Lori's teaching a class, asking you students that are in the class to look at an RNA-seq and pull genes out, make a hypothesis, and hopefully find a gene that's related to a specific function and phenotype. It doesn't work that often, I hate to tell you. It's very frustrating. You can learn a lot from RNA-seq experiments, but often, you know, the gene that's at the top of your list isn't the one that's driving the phenotype you're interested in. And so we went through that with low oxygen, and we ended up deciding to let the fungus tell us. So long story short, how we came about this is we started screening environmental and clinical isolates. Another tip for students: there's a lot of genetic heterogeneity within the organism that you study. And so we often study reference strains for many years in our respective fields, but there's a lot of cool genetic diversity out there. You can do screens without having to do any kind of recombinant DNA experiment. Just go get a bunch of isolates and screen for phenotypes. And now with genome sequencing, cheap as it is, you can actually track down the mutations that are causative in many cases. But so that was our idea here. So we set up an experimental evolution experiment with a clinical isolate that really struggled to grow in low oxygen environments compared to a lot of the other strains that we had looked at. So we ended up passaging the organism for about 4 months under low oxygen environments. We passage the spores, we collect the whole population in each passage to try and avoid bottlenecks. And we kept passaging. And we ended up, this experiment just kind of has taken the lab in a very different direction than one that we were previously working on, which is what one of the fun things about science is. So we got a strain, and as you might expect, we did recover a strain that became more fit under the low oxygen environments. That was the goal of the experiment. You know, if you're following along, you might predict that the strain is a little bit more pathogenic, right? So if low oxygen environment is important for progressing disease, I actually shouldn't say pathogenic, I should say virulent, a little bit more virulent, okay? So the ability to cause a disease is the same, but the progression of the disease was increased more rapidly, potentially because the strain is a little bit more fit in that low oxygen environment. So those were all great, awesome. The unexpected part that threw us for a loop was this right here. So if you look, here's the strain that we passaged, the parental strain. This is what it looks like in standard lab conditions. Here's the strain that is resulted from the experimental evolution experiment. Hopefully you can see there's a substantial change in the colony morphology of the strain. What was interesting to us is that the other condition where, irrespective of the evolution, changes colony morphology is low oxygen. We've done a lot of experiments, and the low oxygen environment is one environmental condition where we see these morphological changes within the colony. And so we really became curious about if this adaptation to the low oxygen environment was being driven by the change in colony. Was this causative or was it correlative? And so we really dove in and started to investigate this. And in order to do that, we had to quantify the morphology. And so we ended up quantifying the amount of a colony that is vegetative, so not undergoing asexual reproduction. And then one thing that was repeatable was the development of these furrows. So these colonies that were coming out of low oxygen always furrowed. So this rugose colony, the mycology term is a rugose colony. And, you know, surprisingly, if you look in the literature, there hasn't been a lot of, as you know, in bacteria, there's been a lot of work done associating different colony morphotypes with pathogenesis, but in the fungi, less so. So again, functional or just coincidence? So the conceptual switch here for us was that really starting to think about these colonies as communities, and that the structure of the community likely is driving the emergent properties that arise when you have a community of cells together. And so at the end of the day now, we started referring to these. So, mycology, the term is a mycelium. And functionally in the bacterial world, they call these biofilms. So they're communities of cells attached to a surface that produce an extracellular matrix. And so what I'm showing you here is in these colonies of molds, this isn't exclusive to Aspergillus, other molds do this too. They make this beautiful extracellular matrix that helps connect these colonies and these filaments together. And so by any definition, these mold colonies are a biofilm. And again, going back to that CT scan, thinking about the physiological environment in the host that we need to treat. Well, here's an example. Here's a picture now that Josh, where's Josh? He's right here. During his PhD, developed this technique in our lab called FunPact. We're able to clear tissue and then look at the fungus within the tissue. Aspergillus, after it initiates infection, when you're progressing the disease, this is what you have to treat, is these biofilms. So they're adhered to the lung tissue, they're producing matrix, and that's what you have to treat. So suddenly we were like, wow, like we've maybe been studying this from, we often study look at the stress response by inoculating conidia onto a plate. And so are you, is the stress response to the antifungal killing the conidia, or is it actually impacting the hyphae? It can be hard to parse that apart by using spot dilution assays. And so this led to a lot of questions for us. So, first, what can biofilm morphology tell us about the evolutionary pressures faced by Aspergillus in the environment and the host? And then how do these population level scale morphotypes relate to specific phenotypic states that we care about, like virulence, drug susceptibility, and then persistence within a human host or a plant host? And then, of course, I'm a fungal geneticist by training. I love genetics. And so what are the genetic mechanisms that facilitate the biofilm form and function? And then, home run would be to identify small molecules that target these mechanisms, because our current antifungals are really ineffective against the biofilm state of the organism. So just kind of hammer home this point that the environment, thinking about the evolutionary pressures the organism face, when we grow Aspergillus in the lab, the reality is we're growing it in a condition it does not really face much in the environment. It does not see 21% oxygen. Okay? It is in the soil, it is in compost. And so you know that because it conidiates the moment you drop it on a plate. So conidiation generally is a stress response in filamentous fungi. And it is oxygen dependent. Okay? Light and oxygen makes sense. You're in the soil, it's dark, no oxygen, growing vegetatively, but you come to the surface, that's when you want to release your propagules. You have light, you have oxygen. And so in low oxygen environments, we get this induction of these rugose colonies. Okay? It's hard. We don't have an incubator to completely shield out light, but you can get these colonies that are completely rugose, producing very little spores in response to low oxygen environment. And so the strain that we evolved, we ended up calling H-morph because it resembled the low oxygen colonies of the wild type strain. But the reason we gave it a specific name is the H-morph colonies, here's an example right here, they do this when oxygen is present. Okay? So they look like the low oxygen colonies in the presence of oxygen. Okay? And we have the quantitation there. Okay? So we have an H-morph. So N-morph strains can switch between the two different morphotypes, but H-morph are locked into this rugose colony-like state. And so some other ways we've quantified this biofilm morphotype to convince you that they're very different. Sorry, there's a delay between my computer and the Zoom screen. There we go. Okay. So if you want to do genetics, you need quantitative data. So one of the things we looked at, we can use confocal microscopy to set up these submerged biofilm cultures. The nice thing about these cultures now is we don't get asexual reproduction, okay, which we don't see in vivo. And these filaments, they look a lot more like that image Josh took from the lung. So we've kind of really moved heavily into using this model as a way to study mold biology now in the context of infection. But quickly, H-morph, N-morph, you should see some difference, how the hyphae are orienting themselves within the community. And what you're looking at over here is quantification of the angle of departure of each hyphae within the biofilm from the surface. And so H-morph hyphae are growing more horizontal. And with the delay here, I'm a little worried these move. Oh, okay, good. There we go. So the N-morph biofilms, you get this really distinctive, sorry for the computer issue, not sure what's going on, but these really distinctive colony biofilms where the hyphae grow vertically. So one of the hypotheses we have in the lab right now is that is actually aerotaxis. And so we're hoping to figure out what the oxygen sensor is. Is it actually aerotaxis? There could be other nutrient gradients that are forming that are driving this as well. But that's our favorite hypothesis right now. And then if you look at the H-morph colonies now, hopefully right away you can see there's a lot more space between the hyphae. And as this thing starts to rotate, hopefully you can start to see that the hyphae are growing, they're longer before they branch. They grow more horizontally, which helps create that space. And so the hypothesis here is these strains now are able to, since there's more mass diffusion capable, since you have more space between the hyphae, the gradients that developing are less steep. They have more access to oxygen. They have more access to other nutrients. And when you quantify biomass in these strains, these guys always grow more. They generate more biomass. Okay, so I'm not going to go through all the genetics. So you're probably wondering what happened in the evolution experiment. So what happened is we found a fungal specific gene that was unstudied in the subtelomeric region of chromosome 5. Like my student's worst nightmare. Are you kidding me? An unknown, unstudied gene that's only in the Aspergillus genus. And so it was, but the good news was it was clean. It was a very clean experiment. There were 3 other SNPs that were high impact after 4 months. And this one was the one. And so it has a nuclear localization signal, which I'm going to show you in a minute. It has this one domain. The secondary structure is a beta alpha beta beta alpha beta motif, which is a nucleic acid binding domain. It could be RNA, it could be DNA. And so it was in a gene cluster that pops up here. I heard Emil was here. Is that right, Emil? Yeah. Okay. So we're working with Emil on this now. So it's in this gene cluster in the subtelomeric region. We called it HAC for hypoxia-associated cluster. This seems to be a regulator of the cluster because if we knock this gene out, the other genes in the cluster, their expression turns off. The allele itself that arose, the allele is both necessary and sufficient. So we can take the evolved allele, put it into wild type strains, and we get this beautiful rugose morphology. But you also can just take the wild type allele, which is silenced in the wild type strain that we worked on, and overexpress it at an ectopic location and get the beautiful morphology itself. So what happened with the allele actually increased the expression levels of the transcription of what this gene is. And we don't know why. It could be transcript stability. It could be increased translation rate. We're not sure. But you increase the levels of this gene called HRMA now. You induce expression of all the genes in the cluster, which is great. And it turns out there's another gene that we're working on with Emil that was unannotated in the genome. There's a lot of cool biology in these fungal genomes. Don't necessarily believe the annotation you have. If you guys use FungiDB, like take your genome sequence, use RNA-seq, load it up, you'll find all kinds of new genes that are not annotated. I find a new gene every day in our genome. Just that's what I do for fun. At night, I just scroll through. I'm like, oh, yep, there's a new gene right there. Guarantee you will find new genes, and some of them are really important. So here's Emil's stuff that we're working. We just submitted this paper with Emil, and so it turns out that gene cluster is on one of these giant starship elements, and the paper here shows that we found a lineage of Aspergillus where the cluster is moving, and so which is really exciting. We still don't know where the genes come from. They clearly are recently evolved within the lineage. They don't look like they're bacterial, but you know, there's a lot of cool stories about subtelomeric gene expansion and parasites, for example. Malaria, the reason you don't have a malaria vaccine is because subtelomeric antigen variation. It all occurs in the subtelomere region of malaria parasites. They just keep generating new antigenic variants with these new genes. So there's a cool Canada story from Brendan Cormack's group in Canada glabrata, where the subtelomeric region controls adherence to surfaces. A lot of cool stuff. A little bit more about RMA. It goes around the nucleus. We think it's in the nucleolus. We're still working on that. You can see the beautiful localization. It does have that signal. And then what we did is we knocked out the nuclear localization signal. Protein's fine. I'm not showing you the Western blot. But we get rid of the colony morphology if this protein cannot get to that nucleolar or perinuclear region. Okay. All right. So what's driving the phenotype? So the title of my talk was a little bit provocative, cancer-like metabolism. So a couple things. I told you that H-morph occurs when oxygen's around. And so one piece of data that we got, which was really fascinating to us, is that even though Aspergillus is obligate aerobe, this is the parent strain, what we're looking at here is the ability of a biofilm to consume oxygen. You can see very rapidly Aspergillus consumes all the oxygen that's available to it. That's within the biofilm. So the fungus itself is inducing a hypoxic microenvironment in its colony. It's just wild, okay? And then here's our Evolve 20 strain, which is growing more. It's producing more biomass, if you remember from that data, but yet it consumes less oxygen. So there was a disconnect there. That's when we kind of started thinking about cancer a little bit. One of the major challenges, though, the difference between a cancer cell and a fungal cell is all this matrix and this polysaccharide. So fungi make this huge cell wall. We don't have cell walls around our cells. And fungi are decreasing, secreting this out into the environment. And so here's a TEM image of the parental strain. You see that beautiful cell wall. Here's matrix on the outside of the cell wall now. But our H-morph strains, you can see the cell wall is different. It's a lot thinner, and then the matrix is balling up outside. And so it still produces the matrix, but it secretes it away from itself. That may be important in pathogenesis. And we know it's secreting it away because we can stain for beta-glucan, which is what most of this electron translucent layer is. We use a probe, it's called Dectin-1. It is a pattern recognition receptor in your immune system that detects fungal beta-glucan. So just like plants have pattern recognition receptors. They have, chitin's a great one in the plant fungal pathogenesis literature. This is the beta-glucan receptor in humans. So we use it as a tool. We tag a fluorophore to it, and then it binds beta-glucan. You can quantify how much exposure you have of beta-glucan on your fungal cell. So consistent with that, we have that. And so when you have more beta-glucan exposed, the immune system is going to see it more and hyperreact. And so we actually think that that's what's driving the increased disease progression with these H-morph strains. Computer's really slow. Okay, so host damage. So with these H-morph strains, so this is an assay where you're looking at tissue damage based on release of an enzyme when cells lyse in your tissue. And so here's the wild type strain. This is an airway. So the fungus is in the airway, the parental strain. All that black is the Aspergillus growing as a biofilm. And then the purple, those are neutrophils that are coming trying to kill the fungus. But look at our H-morph strain. Look at the airway. The fungus is spread out. It's a whole other part of the paper. Again, the biofilm morphology in vivo and in vitro actually plays out. We don't get those tight hyphae like we do in the wild type. They're actually all spread out. And then here, look at these airways, completely full of neutrophil. And so we think that's being driven by the beta-glucan, which is hyper-inducing inflammation. And then, the animals here basically can't breathe, okay, from these strains. Okay, so how is it doing that? So the metabolic challenge here for these organisms is to make this cell wall. And so what we've done recently is a metabolomics experiment comparing H-morph to N-morph fungal metabolism, and we got a surprise. So in the H-morph strains, anything in red is basically gone. And so what we're seeing here is an increase in pentose phosphate pathway intermediates. And that's important because that's where you get a lot of NADPH from. And as I'm going to show you in a minute, to make a fungal cell wall, you need a lot of NADPH. Okay? And I showed you earlier, when oxygen's low, Aspergillus ferments. Okay, so what we think is happening here is the glucose is flying through glycolysis to generate reducing power, as well as intermediates that are needed to make the cell wall when oxygen is limiting. And so here's beta-glucan. It's easy. So the reason these H-morph strains have more beta-glucan, because you can just take glucose 6-phosphate and shunt it over to beta-glucan in a couple steps. You need a little bit of NADPH, but that's it. There's like one enzyme here, and one there. It's a really simple molecule to make, relatively speaking, for a fungus. The tough one is chitin. Chitin's essential for fungi. It takes glutamine, acetyl-CoA, a ton of NADPH, multiple steps, including the formation of UDP-glycnac, which is important for epigenetic regulation, all other parts of fungal biology to make this. This is a ton of reducing equivalents and carbon that are needed to make a molecule of chitin. Very expensive. Aspergillus makes galactomannan, which is part of the matrix that it secretes. Again, multiple steps requiring NADPH, all coming off of fructose 6-phosphate. So now you have a competition to make chitin or galactomannan. And then the main polymer of the matrix is this molecule called GAG, which requires UDP-glycnac from GalNAc from UDP-glycnac. So again, really a lot of pressure being placed on this molecule to form this extracellular matrix. Okay. And so how does an organism do this? Well, when there's plenty of oxygen, there's no problem. But in these H-morph strains where oxygen is apparently limiting, they found a way to do that. And so one of the cool things that fungi apparently can do is they can do reductive carboxylation in the TCA cycle. So to run the TCA cycle in reverse, okay, to generate that reducing power that you need to make that cell wall, okay? And so when this happens, alpha-ketoglutarate gets carboxylated to isocitrate, okay? And that gets converted to citrate, which goes out. It can be converted to acetyl-CoA, which you need for the chitin biosynthesis. Okay, and the key trick here, the key trick here that tipped us off that this was a cancer-like metabolism was this molecule right here, 2-hydroxyglutarate. It is produced in cancer cells. Okay, and we haven't seen it before in fungi. So what that molecule told us is that in these H-morph strains, the TCA cycle is running in reverse. And we think it's running in reverse to generate NADPH and the intermediates needed to make a cell wall when oxygen is limited. It's a really cool mechanism in order to maintain a structure that you need to survive. Okay. And the cool thing is you get a ton of NAD to keep glycolysis going in that process too, which is absolutely essential. So some of you may have heard about this. So this is the cancer model. So the Warburg effect was discovered in 1924 by Otto Warburg. It is high rates of glycolysis in the presence of oxygen for mammalian cells. The cool thing, and how this exactly works, people are still working. It's a huge area of cancer research, but it provides intermediates to make lipids and nucleotides in mammalian cells. But in the fungus, we think it's providing the intermediates to make the cell wall. And so these H-morph strains are having a Warburg-like metabolism in response to an oxygen deficiency. Now, the question then becomes, that is not answered in the cancer field, and we don't have the answer either in fungi, but maybe we can get there, why the cells get locked in this physiological state. So we have the genetics, and we're trying to figure that out. Why do they stay this way? Why don't they just flip back to regular metabolism? Okay. So a little bit of support for that model. If you actually take these H-morph strains and you put them on media that you are forced to use respiration instead of glycolysis, we completely can change the cell wall, and we completely flip the morphology back to the wild type flat colony morphology. Okay. Just to check, Lori, when's the seminar end? Sorry. 1:10. Perfect. Okay, good. All right. So you may be asking, does this matter in vivo? Well, I showed you the H-morph strain, so glycolytic intermediates. This was Josh's white whale during his PhD. We'd argue about this in my office. So there's a decent amount of literature saying that those respiratory carbon sources that drive respiration were probably the main carbon source in vivo for Aspergillus. So our whole model would kind of be a mess and not relevant to what's going on in the lung if that was the case. Josh was convinced there was glycolytic intermediates available to the fungus within the lung. And so before he left the lab, he made this mutant called PFKA, phosphofructose kinase. You can see it can't grow on glucose, okay? But can't grow on, it grows a little bit on lactate. You can't see it on the slide here. Grows fine on protein, grows fine on acetate, goes fine on glycerol. So it's a great mutant to test his hypothesis. And you know, he unfortunately came and joined Lori's lab before he was able to do this experiment. But I can tell you the new PhD student was really excited because the PFK mutant cannot initiate infection. So the interpretation of this experiment is that glycolytic intermediates are, yes, driving in vivo fungal initiation of the infection. So this glycolytic metabolism is essential for Aspergillus to progress disease. So you'll get one more paper out of your PhD. Okay, this isn't a lab artifact. These are different clinical isolates that we've recovered now from a B-cell malignancy patient, patient with cystic fibrosis, a patient with pulmonary invasive aspergillosis. You can see the really nice H-morph morphology. So this is coming out of patients. And in fact, if you go to the literature, 1975, you can see the first example of this. So again, these are two different patients, two isolates from the same patient. One's H-morph, one's not. We don't know because genome sequencing didn't exist back then if these are related, but we have data now from patients at Dartmouth where we get isolates from the same patient. Here we go. Maybe on my screen. There we go. So these are all isolates from one patient, different colony morphologies. This one is very H-morph-like. These are all related. There's only a couple hundred SNPs between these four colonies. Okay, so in vivo adapting this H-morph-like physiology, which again, we think is because of the low oxygen environment found in the tissue. This provides a way for the fungus to maintain viability, to make its cell wall when it's missing. I think here's about 12 patients that we've looked at with cystic fibrosis that are infected with Aspergillus. And we see H-morph isolates in pink here in almost all the patients. So these tend to be arising in vivo in response to a low oxygen environment. We're using this, I mentioned to you guys early on, you don't necessarily need to do a recombinant DNA-based genetic screen. We've taken these isolates. So here's two from a patient with IPA. There are only 9 non-synonymous high-impact SNPs difference between these two. This was a home run. So this is my new students project. And I think Gillian will appreciate this one. The mutation is an LEA. And so the H-morph strain has a loss of function allele of LEA, which is a methyltransferase Nancy Keller originally discovered many years ago that regulates secondary metabolism and fungal development. And so here's the parent strain. There's nothing wrong with the allele. In the strain that evolved in the patient, we lose this LEA. There's a stop codon that causes a premature stop. And we can do all the necessary and sufficient allele swaps. So here's the parental strain that we work with in the lab often. Here's our control strain, our H-morph strain. Here's the mutant of AF293 from Nancy's lab. It's H-morph, which we went back and looked at her. Oh, there it was. It was in her paper. And then here's the allele from the patient that we put back into the mutant, and it's non-functional. And then if you take the clinical strain that has the loss of function allele and you put the wild type allele back in, we can completely restore the colony morphology. So this is a good one because it gives us a genetic regulator we know something about for how. So the idea here now that this is potentially an epigenetic regulator of that metabolic switch, which is cool because in cancer, a lot of the regulation of the Warburg effect is done through epigenetic modifications in cells. So early days on that, but pretty cool. So just to kind of finish up, I've showed you that these H-morph strains are important in progressing disease. It's not a lab artifact. They're happening out in the clinic. The cool thing is that these H-morph strains have this metabolic rewiring that's driving glycolysis and reductive carboxylation, we think, to really drive cell wall homeostasis. And that in that context, these strains become fit in low oxygen environments and then are able to cause disease and damage to the host. And so I think with that, I will finish up. I was going to show you the drug resistance stuff, but we'll just skip the drug resistance stuff and stop there. Happy to take any questions.
Students first. I'm curious if I'm understanding correctly. Wouldn't it make more sense? I mean, I know this isn't the case. Wouldn't it make more sense for the wild type that evolved in the higher oxygen environment to have like colony morphotype that creates the low oxygen environment to promote growth as opposed to the phenotype, the one that evolved in the low oxygen, creating that?
So the wild type strain,
The one that has the less clear cut, nice looking morphology.
yeah. So I think the answer is this, so I didn't show you, is that those H-morph strains are more sensitive to oxygen. So I think in the wild type strain, if you were to adopt that morphology, so some of the furrowing we see, we think is a stress response to the oxygen that's available in the environment. And so in the reason the wild type strains haven't been selected for this in the environment is because of that oxygen sensitivity. That's our working hypothesis. So like if you take hydrogen peroxide, it just nukes these H-morph strains. And so we think because as you reduce electron transfer through the electron transport chain, you release a little bit of ROS. And so these H-morph strains do that far less. We've measured that. And so we think when you have more oxygen around, when you ramp that up, that those strains would become more susceptible. And so so in the lab, like a 5% fitness defect, you don't think about or care about, but out in the environment, that actually is a huge disadvantage. So that's my hand waving answer.
Cool. Thanks.
It's a nice segue from that question. Do you see a difference either transcriptionally or genetically in oxygen sensors? So globin genes or anything between the H-morph?
Yeah, so good question. So there is a globin-like gene in fungi. It's called fungiglobin. I haven't scooped too many times in my career, but that was one time. So that gene is regulated by the transcription factor that I started at SRBA. And so based on that data, someone pulled it from our RNA-seq and they published a really nice paper on it. But it's really the function, it's a globin, it likely binds oxygen. They didn't show that in the paper, but in bacteria have a homologue. And in bacteria, it's been well studied as a molecule for grabbing oxygen and moving it across the different cytochromes in the bacteria cell membrane. So it's likely doing something like that in fungi, but we didn't see any differences in that gene transcriptionally between our H-morph and N-morph strain.
Thank you.
Off of that also, I'm curious with the morphology of the H-morph strain, do you think that it's just the horizontal growth is just a lack of polarity overall, or is it directionally horizontal?
That's a great question. So I have a postdoc in lab that's super interested in that question. I don't have an answer. But he did his PhD in Rob Arkowitz's lab, who's a cell polarity lab. And so he's super interested in that very question. It's a great question. It could also potentially have to do with the cell wall not being able to support vertical growth. That's another model that we've thought about, that the cell wall is weaker because it can't make as much chitin as the wild type strain, so you can't get that vertical growth. But the polarity question is very interesting.
When you're looking at your H-morph strains, specifically with your metabolomics, do you see an increased production of other virulence compounds, specifically gliotoxin?
Yeah, good question. So if you look at the RNA-seq. Oh yeah, sorry. The question was, do we see changes in secondary metabolite production between H-morph and N-morph, like with gliotoxin? And so with gliotoxin, no, but there are other secondary metabolism clusters that are differentially regulated between H-morph and N-morph, and definitely could be involved in the virulence phenotype. That inflammation could be involved in dictating how the colony organizing itself, for sure. Especially with the LEA result that we got recently in the clinical isolate.
So as a clarification, are the clinical isolates, is it easier for the host to recognize it because there's no matrix there?
Yeah, I mean, Yes, so definitely like, both in vitro with macrophages and neutrophils, like when they see the H-morph strains, the cells activate rapidly, through a dectin-1 dependent mechanism. It turns out that I didn't have time. One of my students, the experiment to do was to take dectin-1 knockout mice and see if that eliminated the virulence of the H-morph strains, because we thought it was all this neutrophils that were being recruited. But like plants, humans have evolved many receptors for these fungi. And so when we got rid of dectin-1, we really didn't see any change in the progression of disease or the inflammation. But there are other receptors of beta-glucan within the system. But the answer to your question is yes, they definitely are recognized. Maria?
What happens if you co-infect, like your H-morph with
Great question. So we've done those experiments in the lab, and it's wild. So you can take up to like 10-to-1 wild type to 1 canidia of H-morph, and after 3 days, the whole colony is H-morph. So we haven't done it in the mouse yet, but we've done it in the lab. I should have shown that. They're really cool. You can do different. H-morph always wins in the lab condition.
Have you ever done the reverse evolution experiment where you take your H-morph and just repeat it in high oxygen?
Yeah, I bring it up quite often in lab. No one's yet taken me up on that one. But it's great question.
I have a question. Do Aspergillus have a AMR problem?
Does it have what?
AMR, antimicrobial resistance.
Yeah, it does. Yeah, I'm sorry I had to skip through the second part. I mean, actually, one piece of data here. Drug data. Here it is. So this is voriconazole and amphotericin B. These are the two antifungals we use in the clinic to treat Aspergillus. And so this is the viability. And you can see the biofilms as they develop become completely resistant to those drugs. And so there are mutations in the target that have been discovered. There are mutations in the drug efflux pumps for the azoles that have been discovered in clinical isolates. And so it is a huge growing issue, particularly with the agricultural use of azoles out in the environment. The story in the field is that is selecting for these target-based and efflux pump-based mutations. Just a quick aside, since it was shocking to me that there hasn't been any crosstalk between ag antifungal development and human antifungal development. Those two haven't talked. And so the story here, it's a sad story. So the azole story is one that's emerging. This is the drug. But the second one is there was a new antifungal in phase 3 clinical trials. It's called alorafim, the DHODH inhibitor. It looks great in the clinical trials. And then out of the blue, it turned out Monsanto had already developed it as an ag antifungal, and they just released it last year for use in the fields. And in the lab, all it takes is like 2 passages on the Monsanto molecule to develop resistance in the target gene in Aspergillus. So so what's going to happen to the human drug is very unclear now. It actually failed. It didn't fail the clinical trial, but the FDA didn't approve it in part because of the resistance issue. So what's actually going to happen there, we're not sure. But it's a great molecule. But so we need more crosstalk between the 2 different groups to make sure we're not targeting the same mechanism, given how quickly these organisms develop AMR.
All right. Well, thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.